Welcome everybody to today's ARI seminar. I'm Fern Hames, I'm the director at ARI, and I'm really delighted to host today's seminar. It's going to be excellent. Now, before I get into what today's seminar is about, I would like to acknowledge that I'm joining you from the lands of the Wurundjeri people. I'm here in Naarm in Melbourne at ARI. I'd like to acknowledge traditional owners, elders past and present, and all the wisdom and knowledge that they bring and the relationships that we aim to share with them. I'd like to also acknowledge all the traditional country on which you're joining us from across the state and beyond. And also acknowledge that it's Biodiversity Month this month. It's September. That means Biodiversity Month for us. It means celebrating all things biodiversity, learning new things biodiversity, and sharing biodiversity stories. So it's a great opportunity for us to share a couple of stories from two ARI staff members today. We've got Ruby and Annique. It's great to hear from them. I know that they have done some conference presentations on this work and got great feedback. It's pretty exciting. It's great to hear about. So it's great for us to have the opportunity to hear these stories here as part of our ARI seminar series as well. There is a bit of an aquatic theme. We've got Ruby talking about fish and floods, and we've got Annique talking about fascinating ear bones. I did want to have an alliterative word to go with fish, floods, and fascinating ear bones, but I am constantly amazed at the information that we can get from otoliths or ear bones. So great stories. But to start, we're going to start with Ruby. We'll hear from Ruby and then we'll jump into questions from Ruby. Then we'll hear from Annique and then questions for Annique or questions from both at the end. If you do have questions, please pop them in the chat and Tess will read those out and help us with those after we've heard from Ruby's talk. Thanks so much, Ruby. Over to you.
Right, thanks, Fern. Okay, so yeah, as you might know, I'm Ruby, and I'll be talking about the impacts of 2022's floods and the low oxygen water that it brought with it on our native fish populations. I'd like to thank all the other authors on this project with me. And firstly, I'd like to acknowledge the traditional custodians of all the country in which we conducted our fieldwork, and I'm coming today from Wurundjeri land, and I pay my respects to the traditional custodians, elders past, present, and emerging, and any other Aboriginal elders from different communities who may be here today. So when we think about floods, we usually associate that with really positive connotations, right? Brings water and life and enriches our landscapes with greenery. You know, it's great for our amphibians. It brings our water birds here. But what happens when it's not so good? Well, we saw in 22 mass floods occurring within our state, causing not only great destruction to our urban environments and our infrastructure there, but also to our native ecosystems. And we saw large fish kills resulting as a result of these floods. And you can see I've got some pictures here of some dead Murray cod and a Murray cod gasping at the surface here. And this was as a result of low oxygen levels within our waterways because of these floods. And I've just got a video here of a fish just to see what this is, the kind of thing we were witnessing throughout this time. Individuals such as this one gasping at the top of the surface of the water, looking for oxygen. And this is a really good indicator of what's happening within our waterways and what we can then do as managers and science people to mitigate the risks associated with floods. So when I say low oxygen water, I'm referring to hypoxic water conditions. But what is that and how does it occur? Well, firstly, we have a lot of rain, and all that flooding inundates the land across next to our rivers and streams. And this then washes all that organic matter back into our stream. So I'm talking about dead leaf material, grass and other things among the landscape, especially if it's near our agricultural farms. So we're bringing in like the pollutants from that area as well. And then once it's back in the water, there's microbes that break down this organic matter, and they do this through respiration, much like us. They consume oxygen and release carbon, and it gets to a point where the dissolved oxygen levels within the water falls to fatal levels that affect fish, so below 2 milligrams per liter. And then this therefore causes fish deaths. And this is what we saw last year across the state. And here I've got a picture here of our beloved Murray cod that has passed away due to these poor water quality conditions. So I was lucky enough to be a part of this project where we were trying to determine the acute impacts of the 2022 floods on key native fish populations and aim to develop some pathways of recovery of where we can go moving forward for next year and to manage floods in the future as well. What did we expect? Well, we expected that native fish abundance will decline among these hypoxic conditions, and that there would be an increased abundance of the exotic species carp, which I've got a photo up here on the top right, you can see, and then poor recruitment of native fish. When I say recruitment, I'm meaning the ability for the young to live to the next generation cycle. So during these floods, you might have had eggs or young juveniles, and the recruitment is what we're looking at is how many of them have survived until they were 1 year of age. And that gives us an indication of how the population is going to move forward, and whether we've lost a whole generation of young fish. And then we're also looking at the recruitment of carp and expecting that that will increase. And here I've got some photos of river blackfish, which is threatened, as well as our Murray cod. We've also got a trout cod here and a macquarie perch, which is very endangered and very beloved. So these are some of the reasons we followed these species, was because they are important both as indicators of our river health, but also socially and culturally as well. So our data collection. We were lucky enough to have monitoring programs that spanned the large majority of the flooding areas. On this graph, you can see where the really intense low oxygen areas are in this light pink area, really along our Murray River. And we categorized our sites into 3 levels of how bad the low oxygen levels were, so high, medium, and low. And we had 184 sites across 17 water bodies, and we looked at 12 species. We then looked at the data before the floods happened. So what was the fish populations looking like? And then we got the data after the floods and did a Bayesian model to understand how these floods have impacted the populations. And just an insight into what our fieldwork looks like. To conduct our fish surveys, we use electrofishing, either backpack here along the stream or on a boat. And the electrofishing temporarily stuns the fish long enough for us to put it in a net, then measure and weigh it, and then put it back in the river, and it's all fine and dandy. So it's a very it's a low invasive technique to survey their fish, and it's quite effective as well. As you can see, sometimes it can be quite strenuous along the river, and it's not always smooth sailing, as I found out in my first year here at ARI. Sometimes you do fall over and you get wet, but it's all a little bit of fun as well when we're collecting these fish. So our key findings. So native fish species reduced in abundance in moderate and hypoxic areas. So in the areas where the flooding was not as intense in terms of low oxygen levels, we didn't find big impacts, which is what we expected. We also saw carp increase in numbers across all the categories, which is alarming. And recruitment declined for some native species, and carp recruitment markedly increased across all categories, which is a significant result that we will be looking at further in the future. But how bad is it? Well, hypoxic events had detrimental effects on abundance of native fish large and small-bodied over a large spatial scale. If you remember the map I had of Victoria and where all our sites were, we saw a lot of declines across those sites. And here I've highlighted just how much the percentage of carp increased, almost 400% increase of population within these areas. And we also saw declines across our native fish species, so almost 100% decline in our populations for trout cod and the Murray-Darling rainbow fish and other endangered species. So we also looked at native species recruitment, which declined. Here we have a young of year Murray cod. And so you can see here that in the green, before the floods, we had quite good numbers. And then after the floods, we saw quite a decline. And that is expected because these young of year fish, they're not as hardy as the adults. They can't withstand the large changes in the environment, and the floods also bring a high velocity of water, which disturbs them as well. So we saw a big crash. This is going to have a lot of impacts going future into these populations, as we won't have as many young fish then going into maturehood to then spawn in the following years. So this is something we will be monitoring in the future as well. On the other hand, we have carp recruitment majorly increasing. So as you can see, before we had sort of smaller numbers, and then afterwards we've just seen a crazy increase in carp numbers, and the population structure of carp as well. It's a large amount of juveniles. And again, we're going to have to monitor this because these young fish are now going to grow up and they're going to become bigger and become a larger problem in the future in our Victorian waterways. So it's really important data. But why are carp such a problem? Like, they're another fish. Do we really need to worry? Well, we do. They are a really noxious exotic species, and they consume our native fish eggs and larvae. And that's a problem because that doesn't, it prevents our native fish from recovering more quickly, because if we only have a small amount of adult fish spawning, and then we have these carp eating what's left there, then we have even less of a chance of our native fish recovering post these floods. Also importantly about carp is their feeding method disturbs the environment. They go along the bottom of the floor, increasing the sediment and turbidity of the water, decreasing the water quality of the environment, and that impacts both our fish, smaller fish, their breathing on their gills, and the vegetation structure within the aquatic environments. And carp are known to live in areas where their water quality is low, and they contribute to that as well. They also outcompete our natives for resources such as insects and vegetation as well. And they produce offspring in large quantities, and they grow really quickly as well. So even though they only produce the same amount as our natives, their young mature much more quickly and are outcompeting our natives for food and other habitat resources as well. They also don't have any natural predators in our system, and that means their population numbers are out of control. There's nothing to control those numbers and bring them down to more manageable levels. So they are a large problem that we need to look at. But what are our recovery strategies? What did we think? Well, connectivity is really important. And this is really important because a lot of our native fish species use water and flooding as cues for their spawning. And so we need to be careful of monitoring our environmental watering, which releases water into our rivers and streams. And this helps both connect populations of different native fish, and that creates genetic diversity within the populations, which therefore makes them more robust to extreme events such as floods and heat waves in the future as well. We also need to look at monitoring our fishways, making sure they're open so we can facilitate that connectivity between our streams, and further monitor our populations through genetic structuring as well. Stocking is proving to be an important way in which we can help supplement our native fish populations and rebuild those numbers to allow more individuals opportunity to breed. And Annique will point out some really important parts of stocking as well in her talk. Also, we need to look at carp removal. The numbers are not going to reduce on their own, so it's important that we can maybe step in and put in some measurements. And again, monitoring is really important. We would not have been able to complete this project if we didn't already have a lot of the monitoring programs that are set up at ARI already. And we were able to complete this project immediately after the floods within the first 6 months of this year and get all this great data. And I'm able to present this to you now because of the hard work and the monitoring programs that are supported by our staff here. And we just want to continue to be able to do that. I've looked at the acute impacts today, but we also have been needing to look at the longer term impacts in the next couple of years, especially as we move into El Nino. And so my final key points: hypoxic water causes declines in key native fish populations, and exotic species carp increased substantially post the 2022 floods. And you can find me at Diga. And that's it. Thank you.
Ruby, thank you so much. That was terrific. And you know, it's just, I feel a bit kind of defeated in a way. It's not a great thing to reflect on, but I'm aware that we're going to have more extreme events, and understanding how we respond to these things is going to be even more important as time goes on. And I reflect on, you know, for a while we used to call carp European carp, but you know, now we call them common carp because we're reminding ourselves that they are very common, and they originated in China in places that had high temperatures, low flow, high flow. Like, they deal with extremes really well. So as we're going into a climate with more extremes, we're going to have to deal with carp even more. So grateful for all the work that you're doing, helping us understand what this means and how we can give native fish the upper hand to support these systems. Now I'm going to have a quick look for questions. I think, Tess, you might have some questions if they're not in there yet. I think you had some that people had sent in as well. So what would you like to ask, Ruby?
Well, Ruby's actually answered a couple of the ones that I prepared earlier, but a couple of questions. Is there anything we can do to reduce or prevent hypoxic events?
Yeah, so that's a really good question. It's important to note that these floods were caused by the large amount of rain and flooding through our waterways, which brought in a lot of the organic matter. Hypoxic or low oxygen water conditions can also happen during really low flow events and increased temperatures. So there's a few ways in which we can help reduce the chances of this happening. Environmental watering is one of those ways, where we release some water into the system, and that helps bring some fresh water with oxygen in it, flushes the system out, and provides some relief for those habitats and those fish. We can also do some other things to reduce the impacts. For instance, ARI did some fish rescues, which can help some of the fish, but it's quite hard in these, you know, the floods are such an extreme event, so they can be quite hard. There's also physical aerators that people have looked into, but mainly environmental watering and monitoring how our rivers are going at each site, so we can see whether they're dropping really quickly in terms of oxygen levels, or are we going to have a long period of high temperatures, and what that means for the oxygen as well.
Sorry. So given it has something to do with, you know, material washing into these waterways, particularly in flood events, does that mean that, you know, vegetation along river banks and things like that might stabilise that might be beneficial?
Yeah, it is really beneficial. I mean, always native vegetation along our riverbanks are really important. Not only do they stabilize a riverbank so that the sediment isn't creating a lot of turbidity in the water, and that is really bad for fish in terms of their breathing through their gills as it smothers them. And what little oxygen there is in the water is even harder for them to take in. So that's important too. But vegetation also creates a lot of habitat and food availability. For eggs and larvae, the vegetation is really important for the smaller fish to be able to withstand some of those intense events as well. It gives them some protection. And the vegetation as well, they produce oxygen, just like or trees or any other plant. So having those within the water system is always beneficial to help mitigate the impacts of the microbes consuming the oxygen and releasing the carbon. So, yeah.
Yeah, awesome. I've got a question from the chat. I'm not sure if you know anything about this particular project. Jared's just answered it in the chat anyway, but there's a question about whether the ARC research project on dotterless carp that was designed to restrict reproduction, whether that worked or not. Do you know much about that?
Yeah, I don't. That was probably before my time at ARI. But it's interesting to note, to get different ideas of how to control carp populations. Fishways is one of them, where we can prevent carp from invading different areas in intense numbers. But it's really hard. It's not like foxes or cats where you can trap them as easily, it's because the rivers are all connected, so it's quite a difficult task, and this is why they've become such a prolific problem across the world. And it's not easy, but we're looking into suggest ways in which we can do this.
I think that's it for the questions. We've got some comments from the chat as well. But yeah, thank you so much, Ruby. Fern, did you have any other questions?
No, I was just reflecting on the dotterless carp thing. I think one of the issues was that to have any kind of impact, it was going to take many, many decades. And there are more immediate ways that we need to deal with it. And Jared's, referenced some of the methodology, which was not that successful. So, it was good to explore. It's always good to explore these things. That one didn't look like it was going to have the success that it did. It's interesting, Ruby, you're talking about fishways. One of the things that I think was really, innovative and an exciting solution was the Williams carp cages that we put in fishways to sort carp out from native fish. And if people have seen those operate, I think they're such an elegant idea, because they just, we know that carp jump and native fish don't. So they literally sort carp out from native fish just through that jumping mechanism. It's such an elegant, beautiful idea to, sort carp out. So you can sort them out, but then the issue, of course, is what do you do with tons of carp that are in some remote area or somewhere that's, a little bit hard to deal with them. But it's exciting when we see those kinds of innovative things develop, and they can always trigger other ideas. Well, what could we do with that? And what's something else we could do? So it's a continually evolving area, and I'm grateful to people like you who are going to keep working on this and come up with some extraordinary and innovative ideas. So thank you. Thank you very much. All right, perhaps we'll, and if you, please, people in the audience, if you have more questions or comments about this, put them in the chat and we'll come back to them after Annique's talk as well. Annique, over to you. We're going to hear from you about how amazing are fish ear bones.
Good afternoon, everyone. For those of you joining from outside of the Arthur Riley Institute, I'm Annique, and I'm part of the Applied Aquatic Ecology team, which support and guide sustainable ecosystem policy and management to ensure healthy ecosystems. Today, I'm going to share with you how a fish's ear bone can reveal some neat insight into guiding management decisions relating to recovery actions. I'd like to acknowledge the traditional custodians of the land on which I'm currently on, the Wurundjeri people, and the traditional custodians of the Victorian waterways throughout the Murray-Darling Basin where this research was carried out. I pay my respects to those traditional owners and the elders, past and present, and Aboriginal elders of our community who are joining with us today. Before I start, I also want to acknowledge the following group for their support throughout the life of this project, particularly the Murray-Darling Basin Authority and Victoria's DECA VEFMAP program for their funding and continued interest, and of course the team at ARI for their hard work in bringing this research together. So my focus will be on the use of juvenile cod earbones, which are called otoliths, as a means to determine birth origin, that is, to identify whether a fish from a specific hatchery has been stocked from a hatchery, from a fish from a specific waterway has been stocked from a hatchery or spawned naturally in the wild. And we're aiming to enhance recovery action through these answers. First of all, let's understand the significance of Murray cod in the realm of conservation. So the Murray cod is an iconic species in the Murray-Darling Basin. It's the southeastern Australia, this little yellow area here. And it has immense conservation, cultural and recreational value. Murray cod is the largest exclusively freshwater fish in Australia and can grow up to 1.5 metres. It's been aged up to 49 years of age, I do believe. And while historically abundant, Murray cod has displayed reductions in their range and abundance recently. And as a result, they're currently listed as threatened in Victoria and vulnerable nationally. So some of these threats to Murray cod are presented in a number of ways. Some of the major players are flow modification, habitat alteration, barriers to movement, and the introduction of exotic species like Ruby was talking about, carp. The substantial decline of Murray cod as a result of these threats, in addition to their conservation value, is the reason why they're often a key objective in a range of recovery actions, including environmental flows, habitat restoration, and stocking via captive breeding. So environmental flow being one of the biggest conservation intervention investments in Australia. EFLOW investment is worth about $1 billion a year. Environmental water delivery is managed to improve and maintain the health of our waterways and wetlands, including the aquatic life that depends on them. Now, we identified a gap in the knowledge here because these recovery actions aimed at boosting fish recruitment are often undertaken together in an integrated manner, which means it's difficult to determine what is and what isn't working. More specifically to recruitment, we have monitoring programs in place to evaluate how these interventions are faring, but a shortfall in many of these programs is that they can't define native origin. So for example, environmental water is used to enhance the breeding success of Murray cod, amongst other things, and stocking is used to bolster fish numbers. So we have a monitoring program that goes out and catches a bunch of juvenile fish, but all we can say based on this data alone is that one or both of these has worked. And so understanding whether these are stocked fish that have survived or in situ recruits that have been spawned naturally is critical information here. So to provide better support in evaluating outcomes of these recovery actions aimed at boosting recruitment, we turned our focus to differentiating stocked fish from wild fish by performing microchemistry on the otolith of juvenile Murray cod. Now there's an image of a Murray cod earbone here prepared for microchemistry analyses. And otoliths are commonly used to age fish using a method much the same as aging a tree, cutting a cross section and counting the growth rings. And you can see the growth rings on the tree here, as well as the Murray cod otolith. So when fish lay down these growth rings, they also lay down chemicals from the aquatic environment, creating a chronological record of the environment experienced throughout the fish's lifetime. And when microchemistry is performed on the core and edge of the otolith, these values produced are known as chemical signatures. And they're used as location markers to work out where a fish was born. So the core signature of the fish represents the location of birth, and the edge represents the location of capture. To determine whether a fish collected from a waterway has been stocked or spawned naturally, it's as simple as seeing whether the core signature of that fish is a match with either the signature of the hatchery over here, it would be stocked, or the waterway it was captured and it would be naturally spawned. So from this plot on the left here, you can see distinct chemical signatures of the 5 Victorian waterways from the Murray-Darling Basin highlighted in this map. And we have the 4 hatcheries which stocked the fingerlings into these waterways. And these were used as location markers in our birth origin analysis. Some waterways showed more distinct signatures from the hatcheries than other waterways. So, for example, the Broken River highlighted on the left was quite easily distinguished from all the other hatcheries, which are highlighted on the right, whereas the Campaspe River, highlighted third from the left, is very similar to the 2 that are highlighted there, MDF and Snobs Creek. And so from a birth origin analysis, fisheries managers would be better off stocking the Campaspe with one of these 2 hatcheries, Barriers or Silverwater. I've got to say these particular waterways were ideal for our research because they received both stocked fish and environmental water each year. So once we have the signatures for each waterways and hatcheries, we can move on to identifying whether fish captured from these waterways are stocked or spawned naturally. And each system displayed varied birth origin results. You can see the Broken River here shows all natural recruits. As you can see, the core signature for the Broken River juvenile Murray cod in the centre here matched really well with the Broken River signature, also in red. The Gumbower Creek, however, showed a mix of both naturally spawned recruits and stocked fish, which is a good indicator for fisheries managers that stocking is working in this waterway. So how can this information help us? It's a big question. I'll run through a case study which focuses on one of these waterways, which is the Midda, and we can check out some interesting results we uncovered using birth origin analysis that we were able to determine the conditions that led to this increased stocked fish survival, as well as in situ natural recruitment in Murray cod. So like many fish occupying modified ecosystems, as a result of water regulation, following dam construction and operation, Murray cod were actually thought to have disappeared in the Midda, this reach here in blue, due to severe alterations in the river's thermal profile, which is influenced by cold water pollution. It comes about from water release from dams. So in this plot on the left, the chemical signatures show the Midda results, and we've got the hatchery signature. And in the centre here, we have some clusters which represent the natal signature or birth origin of Murray cod juveniles that were captured in the Midda in 2021. And you can see in this middle cluster, there are 2 distinct clusters of natal signatures. The higher values match well with the Midda signature, and the lower values fell nicely into place with the hatchery signatures. Now that's showing survival from this cohort of both stocked and natural recruits. And this was the only year with natural recruits survival in this reach of the Midda, which coincided with no cold water pollution during the Murray cod breeding season, which is late spring, early summer. Also of interest in this particular year was the improved survival of stocked fish, which coincided with periods of limited flow releases during summer to early autumn. And these kind of results are really valuable at highlighting conditions, the conditions influencing juvenile survival must also be considered in addition to those associated with spawning when assessing important drivers of recruitment strength in Murray cod. Yeah, so examining birth origin of juveniles captured across these several years from this reach, we were able to document the influence of cold water pollution on recruitment success. And this influence cold water pollution was found to have on recruitment has since been embedded in the environmental guidelines for water authorities, which also has implications for stocking. And that is on occasions when water doesn't have to be transferred out of the lake during November, don't do it, and you'll get natural recruitment. And in the low transfer years in summer, if you coincide your fish releases during the times of minimal summer releases, your stocked fish will survive through the early critical life stages. So in summary, I mean, as you can see, birth origin analysis has proven to be a really powerful tool for us to guide decisions in enhancing recovery actions. It's given us the capacity to provide recruitment information to support river managers in understanding their restoration actions, which involve like these immense intervention investments, such as environmental flow, conservation stocking, and habitat restoration. We've also been able to provide that much needed information to fisheries authorities relating to their current large investments in fish stocking programs. By identifying birth origin of these stocked juveniles, we can evaluate the effectiveness of these programs and, you know, make better decisions down the line about the most suitable waterways for future releases. And so I guess through incorporating birth origin information into recovery actions, stakeholders can work towards more effectively preserving fish populations, maintaining ecosystem integrity, as well as ensuring sustainable fisheries for our future generations. And that wraps up things from me.
Thank you, Annique.
No worries.
Applause all around. Woohoo! That was fabulous. I'm just amazed at what we can learn from odorless. It is just extraordinary, really, isn't it? It's these tiny little things. You know, I remember having some as earrings once. They're tiny, you know, and we can get so much information from them. It is astounding and quite exciting and really evolving.
Yeah, I couldn't agree more. And you know, not only birth origin, but migration and aging, and there's a whole lot of wealth of information hidden in there.
Yeah, we can track where they've been. It's amazing.
Yeah,
incredible. Tess, I'm going to let you manage questions. I think Robert's got his hand up, and nice to see a question in there from Charlie. It's great to see a whole lot of familiar names on the list today. But yeah, perhaps Tess, you want to see what Robert's got to ask?
Yeah, if you could pop the questions in the chat, I'll be able to read them out. But I've got a couple of questions pre-prepared while you do that. I was curious about what the cause for the different colour banding is in fish ear bones.
Yeah, that's an interesting one. So because they go through like different environmental changes, temperature, for example, and food availability, it can lead to more pronounced zones. So if you have warmer water and an increased food availability, for example, then you'll have the wider, lighter bands. And then in the cooler water, it's less productive, often in those winter months, and it will lead to the narrower, darker zones. Another little fun fact, which is aligned with that, is the width of those zones or bands is proportional to the fish's growth each year. So you notice, like in the center of the ear bone, they're quite wide when they're growing a lot in their younger years, and then as they get older, those bands get narrower.
Yeah, wow. So they really are kind of like trees.
Yeah.
So my other question was, coming from someone who hasn't had much to do with the water space, was, you know, what are the kind of the different reasons that we release water down rivers?
Yeah, there's loads. So I know that Lake Dartmouth, where this study was focused, that was constructed to achieve irrigation transfers, so for agriculture downstream, as well as hydroelectric generation. So water release from a dam, it flows through turbines and it's converted into energy as the falling water into electrical power, which is a great source for renewable energy. But I also know through some field work that's been delayed this year because there's been so much rain around, like it's also like drought control, flood control. Sometimes you just need to like create a bit of airspace in the dams to prevent downstream flooding. Obviously environmental flow, many systems require that minimal flow to maintain the downstream and protect the aquatic ecosystem. Yeah, so there's I mean, community water supply, industrial use. It just depends, I think, on the purpose of the dam.
Yeah, thank you. And then the other thing that I was wanting to ask was, is there any way to protect native fish from the temperature fluctuations that happens as a result of these releases?
Yeah, it's a tricky one. Ideally, water releases which pose, you know, a threat to cold water pollution should be timed outside of the native fish breeding periods, which is spring, early summer, to increase those chances of successful recruitment. But realistically, that's not always feasible when our irrigators, for example, need large volumes of water at those particular times in the warmer months. So potentially, more gradual releases to meet the water demands in some cases could assist to avoid those rapid temperature changes. As I mentioned in my talk, the regulations and policies is a great option. Just regulations that require dam operators to prioritise the protection of fish and aquatic ecosystems when managing those temperature changes. And there's another one, I don't know how popular it's been, but the selective withdrawal of dams. So that allows you to, for water to be drawn from the dam at different depths. So you can access water at varying temperatures, which might be closer to the natural downstream temperature. I know Lake Dartmouth temperature, because it's got the thermocline, which is you've got your warm water at the top, and then the colder water sits down the bottom. And if your outlet pipes are right down the bottom where that cool water is being pumped out downstream, they measured a temperature difference of about 12 degrees at some stages. So, you can understand how that can have an impact on the fish and their recruitment and the viability of like their eggs if they're not able to sustain water temperatures below 15 degrees.
Yeah, wow.
Yeah.
So
Tess, I reckon this is really relevant to Lisa's question in the chat.
Ah, yes.
Can you see that? It's really relevant to this conversation about temperatures and flows and timing.
Yeah, could we be unintentionally impacting on recruitment with all the releases of environmental water in spring and summer as well?
Yeah, the environmental water, I think it's so gradual. I think that we would do that more mindfully. I think it's like these big volumes of water that are being dumped down for irrigators. I think there's been a fair bit of research around that to protect, not to change the temperature that dramatically.
Yeah, and the other part of the question from Lisa is, do we have an idea of the temperature thresholds that the fish have?
Yeah. So one study, we know that at Murray cod eggs, 14.8 degrees, you'll get 50% larval mortality. At 13 degrees, it's 100% larval mortality. You can't survive. And so ideally, if you can keep the water, above 15, then, you're going to potentially be doing a lot less damage.
Yeah, and Ashley's just added to the chat. Yeah, Robert, if you could please put your question in the chat. Unfortunately, we can't, in the presentation mode, we can't hear you ask your question.
Yeah, sorry, Robert. We're not ignoring you. It's just the way that the audio works with this particular version of Teams. We can't open it up, but please write it in. That would be terrific. Thank you. And thanks, Ashley, for prompting us on that one. Just going to go back up and see if there are any other questions in the chat. Tess. Oh. So, Annique, this is that moment when you get the opportunity to say the thing that you didn't have time to say. There's always, you've got, I've only got a few minutes, but I'd like to say all these other things.
Look, I don't know if everyone else knows all about the other the benefits that we use odorless for. So not only is it birth origin, it's also aging. Obviously, you can see the aging of the rings, but another really cool technique that we use odorless for is checking out their migration and how they move and how they respond to environmental stresses. So as I mentioned, the ear bone, from the centre to the outwards, that's the start of its life, and this is the most recent part of its life. And you can get transect samples of the ear bone and then send them off for microchemistry analysis, and it will tell you where you can match up those signatures of like where they've moved through the creeks and the rivers throughout their lifetime. So yeah, it gives you a really neat snapshot of their life, and you could potentially, pair that up with, okay, what's happening in the environment? Like why did so many fish move down into here? And yeah, it gives us a lot of insight into, are we doing the right thing or could we do things better?
Yeah, including whether they respond to environmental flows.
Yep. Yeah.
Yep. Such an incredibly useful tool. Lisa has another question there, but I don't know the answer to this. I don't know if you do.
Oh, no.
Yeah, so Lisa's asking whether we know whether 1 billion per year for the environmental flows was arrived at. We probably don't know.
I have been given that by one of the head seniors in the team, and I'm assuming a lot of it would be, I mean, water is an expensive resource, and so there's a lot of it is probably adding to that. But I don't know the entire breakdown of it, I'm sorry.
We do know that water is incredibly valuable and becoming even more so. So it's crucial that we have deep understanding of how and when to use it in the best possible way.
Yeah, that's right.
Yeah. Okay, last call for questions.
Yep.
We
Put
might
them in
have
the
the
chat.
opportunity. Yeah, put them in the chat. We may have the opportunity to give you a few minutes back in your day, but we're also super interested in hearing your questions. While we're just looking for that, I will thank everybody today, all the speakers in particular. Big thanks to Ruby and Annique for coming along and sharing your stories. That was fantastic. Tess for doing the tech today. Everybody who came along in the audience, particularly those who asked questions. And yes, Marion, in fact, indeed, we do, well, not still running, but we did run an amazingly interesting citizen science project getting recreational fishers to donate their otoliths, particularly for golden perch and Murray cod. Chat with Pam Clooney if you want to know more about that one. But yes, absolutely. If they're going to be taking fish anyway, it's a great opportunity to have a look at those otoliths and see the stories that they tell us. Annique, did you want to add anything more about that that project where we got the fishers to send in the otoliths?
No, I haven't worked on that directly, but is this the golden perch project? Yeah. It's helped us just get a bit of, like, citizen science is always brilliant. I'm such an advocate for it. So yeah, continue sending those in, and if the fish is already, unfortunately with the ear bones, you have to kill the fish to get the ear bone, and so it's brilliant if we could further use the recreational fishing skeletons for more science, then all for it.
Yeah, and it is a bit tricky to get an otolith out, so just the head will do.
Yeah, we can sort it out.
Yeah, we can help. Yeah, just great to get every bit of information that we can from every fish. Oh, should we keep going? Paul O'Connor's got a question about whether crucian carp in the broken cod were feeding on them. I don't know. Annique, do you know about that? Well, Ruby, do you know about that? That's a Ruby question. Ruby.
Yeah, I'm not sure about that actually. With our carp data, we often, when we're taking our surveys, we're getting huge amounts of carp. So I'm not sure whether we looked in closer, there would be crucian carp there in our data frame. But yeah, potentially. I'll have to ask the guys who went out and did that field work whether they also saw any of that. But that's interesting that, cod were feeding on them in December. Yeah, I'll look into it. I'll ask around. Thank you. Thanks for the question.
That's one of the beauties of giving native fish the upper hand, right? They can feed on some of these things. Oh, thanks, Paul. Good on you for sending the video.
Yeah. Oh, Zeb hasn't shown me yet, so.
Excellent. Now you know what to do. Go and ask Zeb. All right, I might finish it off there. Thanks so much